The study, Improving the Efficiency of Rubisco by Resurrecting Its Ancestors in the Family Solanaceae, published April 15 in Science Advances. The senior author is Maureen Hanson, the Liberty Hyde Bailey Professor of Plant Molecular Biology in the College of Agriculture and Life Sciences. First author Myat Lin is a postdoctoral research associate in Hanson's lab. The technique allowed the scientists to identify promising candidate enzymes that could be engineered into modern crops and, ultimately, make photosynthesis more efficient and increase crop yields. Researchers used tobacco as the experimental model for their studies of Rubisco. We were able to identify predicted ancestral enzymes that do have superior qualities compared to current-day enzymes, Hanson said. Scientists have known that they can increase crop yields by accelerating photosynthesis, where plants convert CO2, water and light into oxygen and sugars that plants use for energy and for building new tissues. For many years, researchers have focused on Rubisco, a slow enzyme that pulls, or fixes, carbon from CO2 to create sugars. Aside from being slow, Rubisco also sometimes catalyzes a reaction with oxygen in the air. By so doing, it creates a toxic byproduct, wastes energy and makes photosynthesis inefficient. Hanson's lab had previously tried to use Rubisco from cyanobacteria, blue-green algae, which is faster but also reacts readily with oxygen, forcing the researchers to try to create micro-compartments to protect the enzyme from oxygen, with mixed results. Other researchers have tried to engineer more optimal Rubisco by making changes in the enzyme's amino acids, though little was known about which changes would lead to desired results. In this study, Lin reconstructed a phylogeny, a tree-like diagram showing evolutionary relatedness among groups of organisms, of Rubisco, using Solanaceae plants. By getting a lot of genetic sequences of Rubisco in existing plants, a phylogenetic tree could be constructed to figure out which Rubiscos likely existed 20 to 30 million years ago, Hanson said. The advantage of identifying potential ancient Rubisco sequences is that carbon dioxide levels were possibly as high as 500 to 800 parts per million, ppm, in the atmosphere 25 million to 50 million years ago. Today, heat-trapping CO2 levels are rising sharply due to many human activities, with current measurements at around 420 ppm, after staying relatively constant under 300 ppm for hundreds of millennia until the 1950s. Lin, Hanson and colleagues then used an experimental system developed for tobacco in Hanson's lab, and described in a 2020 Nature Plants paper, which employs E. coli bacteria to test in a single day the efficacy of different versions of Rubisco. Similar tests done in plants take months to verify. The team found that ancient Rubisco enzymes predicted from modern-day Solanaceae plants showed real promise for being more efficient. For the next step, we want to replace the genes for the existing Rubisco enzyme in tobacco with these ancestral sequences using CRISPR, gene editing, technology, and then measure how it affects the production of biomass, Hanson said. We certainly hope that our experiments will show that by adapting Rubisco to present-day conditions, we will have plants that will give greater yields. If their method proves successful, these efficient Rubisco sequences could be transferred into crops such as tomatoes, as well as those from moth. The study was funded by the U.S. Department of Energy. Materials provided by Cornell University. Original written by Krishna Ramanujan, courtesy of the Cornell Chronicle. Note: Content may be edited for style and length. Myat Lin, Heidi Salihovich, Francis K. Clark, Maureen R. Hanson. Improving the efficiency of Rubisco by resurrecting its ancestors in the family Solanaceae. https://ift. tt. T19 Juas. Accessed April 18, 2022. Thanks for watching. Please subscribe my channel for more videos.